Good afternoon, everyone. My name is Joanne Kwong, Columbia College Class of 1997, and co-chair of the Intellectual Programming Committee and executive board member of the Columbia College Alumni Association, or CCAA. The CCAA seeks to represent the diversity and richness of alumni, always with a deep connection to current students. Through intellectual programming, networking, and community building, the CCAA provides opportunities to stay involved, have an impact, and support our students and fellow alumni. Our program today is presented to you by the CCAA. I'm here today with my intellectual programming co-chair and partner in crime, Kurt Roloff's class of '88, and director of alumni relations, Thandi Jarrett Abdulali, as well as many of our hardworking committee members who meet throughout the year to create interesting programming for all alumni, both in person and virtual, so that we can continue to share intellectually curious experiences together, even if we're far away from those days in Butler or on the steps. Today, we have a big topic, Short Take, which is a virtual series of short lectures on a variety of topics by our esteemed faculty who help us to understand and break down big issues in the news that relate to their research and often incorporate texts and readings from the Columbia curriculum. Today's presentation is titled Emotional Brain Development and the Role of Parents. It is our first in a series over the next year related to the overarching theme of wellness, which is much needed during these times. We are so excited to have with us Professor Nim Tottenham, who is a Barnard alumna from the class of 1996, my era, and who I personally will always remember as performing in each year's varsity show. After teaching at UCLA and Cornell, Professor Tottenham returned to Columbia in 2014 as a professor. She now chairs and is a professor in the psychology department and director of Columbia's Developmental Affective Neuroscience Laboratory. Her research examines the development of emotional behavior and associated brain maturation, including the powerful role that early social experiences have on the brain. She has authored more than 140 journal articles and book chapters and is a frequent lecturer both nationally and internationally. She is a member of the American Academy of Arts and Sciences and a fellow of the Association for Psychological Science and the Society for Experimental Psychologists. Her scientific contributions have been recognized with the National Institute of Mental Health Brains Award in 2010, the American Psychological Association's Distinguished Scientific Award for Early Career Contribution to Psychology in 2015, and the National Academy of Sciences Troland Research Award in 2020. And without further ado, we are so excited to give a hearty welcome to Professor Nim Tottenham.
Great. Thank you. Thanks, Joanne. I'm going to share my screen. Okay, great. Well, hi, everyone. Welcome, Columbia family. Thank you for taking time out of your day to talk a little neuroscience. Today, I'm going to be talking about our emotions, how they're built, how we learn to control them, and what parents are good for anyway. So let's start by defining what we're talking about. We're really going to be focusing on our ability to regulate our big emotions. And regulating our emotions is very difficult. However, it turns out humans are the most emotionally regulated species in the animal kingdom. However, it takes us a long time to get there. So if you think about the ability of a 2-year-old to calm down big emotions that they're having, they're going to have more difficulty than a 20-year-old. And there's going to be a lot of individual differences. So some people are going to end up being very skilled at regulating their emotions, and others are going to struggle a little bit more. So to understand how the system constructs itself over the first 2 decades of life, we need to get into a little bit of neuroscience. And I'm going to be focusing on 2 brain regions throughout this talk. And to orient you, we have the front of the brain here on the left and the back of the brain here on the right. And I want to draw your attention to the amygdala, which is a cluster of cells that sits deep in the center of the brain. And the amygdala is responsible for our emotional learning and our emotional reactivity. So a simpler way to say it is if you've ever been at home alone at night and you hear a creak down the hallway and you feel that all is not right with the world, that's your amygdala screaming. Now, the amygdala is really good at helping us discern safety from danger. In other words, the amygdala has helped keep us alive as a species. However, we don't want the amygdala active all the time because that sets us up for a number of mental health problems. Now, in adulthood, we have connections between the amygdala and this big swath of cells called the medial prefrontal cortex, which sits right behind our forehead. And we can think about these connections as being a superhighway between the amygdala and this medial prefrontal cortex. And these connections help the medial prefrontal cortex regulate the amygdala or calm down the amygdala. And the nature of this neurobiology is important because it produces a lot of our emotional behaviors, a lot of those individual differences in our emotional behaviors that we see in adulthood. And a lot of the work that our laboratory focuses on is trying to understand how early experiences we have, in particular early social experiences, how they contribute to the construction of this neurobiology, so that we can understand why early experiences that we have can have such an outsized impact on our emotional behaviors later on in life. So to show you how we study the developing human brain, I have an image here of my daughter, and she is in an MRI scanner. And she's wearing video goggles here where she can watch a fun cartoon, for example, SpongeBob SquarePants. We play her the sounds that she's going to hear so she can practice. So I'll play that for you now. So once she's feeling comfortable, we can then change out the video and present to her images that we know are really good at activating the amygdala. And an image that works really well for us is another person's fearful face. So we can show these types of images to participants across the first 2 decades of life. And what we found is that the amygdala develops very early. It's apparent very early in childhood. Not only is it apparent and ready to go early in childhood, but it's extremely reactive, more so than it is at older ages. And that makes a good bit of sense because the amygdala's job is to learn about the safety and danger of the world. And when we're little and relatively new to this planet, we have a lot of learning to do about the safety and danger of the environment. So the amygdala develops early. Now, what about the prefrontal cortex? Well, I'm showing you here a video of the brain growing up. The front of the brain is here and the back of the brain is here. And blue color is going to indicate reaching adult levels. So if I play this video, what you can quickly see is that the prefrontal cortex is one of the latest regions to develop. And in fact, current data estimates that the prefrontal cortex reaches maturity by about 25 years old, which of course, car rental companies knew before neuroscientists, which is why they charge more money for people that are under 25. So the prefrontal cortex seems to be developing late. What about those connections between the prefrontal cortex and the amygdala? So to describe the data, I have to introduce a metric we use called functional connectivity. And in brief, what we can do is we can extract the signal from one region, like the amygdala, and plot it over time, and do the same for the medial prefrontal cortex and plot it over time. And the degree to which they're correlated with each other or they're talking to each other gives us an indication of functional connectivity. So now if we look at how functional connectivity develops over time, we see in adulthood, I'm not showing you activity here. The colors will represent the strength of those connections between amygdala and prefrontal cortex. You can see in adults that the amygdala has strong connections with the medial prefrontal cortex, indicated by this orange blob here. But that orange blob is notably absent in children. So we're seeing a slow maturation of those connections between amygdala and medial prefrontal cortex. So if we put all the data together, we see a picture emerging like this. With age on the x-axis, we get strong early amygdala function, but that's happening in the relative absence of those adult-like connections with the medial prefrontal cortex. And that raises a very interesting question for us and makes us look closely at the first decade of life to ask what is regulating the amygdala during a time when prefrontal cortex is not quite ready to do that independently. And to answer this question, we should appreciate that, of course, the amygdala is not developing in a test tube in isolation, but it's developing in a very special environment, a species expected environment, which is the parent or the primary caregiver. And of course, this is a universal for humans. And when we talk about the primary caregiver or the parent, we could be talking about the mother, we could be talking about the father, grandparents, adoptive parents, older siblings. And the point is that this attachment relationship is a learned relationship. And much of the work I'll be talking about today is that process of learning the attachment. Now, humans are not alone in spending time with our caregivers. But we do enjoy some outsized status. We do enjoy some outlier status in how long we spend with our caregivers. So if you think about an animal like a kitten or a puppy, they stay with their parents for maybe a few weeks or a few months, and then they can be independent. But for humans, we stay hanging around our caregiver for years or decades, depending on the structure of your family. And that's shown here in some of these pictures on the left. And I've always been very fascinated by that because it's a huge energy suck for a parent to raise another human being for a couple of decades. And so this begs the question, why would Mother Nature build us this way, where parents are expending a lot of metabolic energy to raise another human being? And one of the arguments that we find very compelling is that this prolonged period of childhood in humans affords us this luxury of having this external caregiver who can scaffold brain development, much like the scaffolding on buildings directs the nature of the way that that building is constructed. A caregiver can provide scaffolding to direct the nature of development. And that affords us this really long period of brain plasticity. And brain plasticity is simply the ability of the brain to change in response to learning. And so, in other words, humans are able to be such magnificent learners and become such sophisticated creatures because we have this long period of caregiving. And so I just want to show you this quick movie of what a plastic brain looks like, because often when we look at pictures of brains or we think about a brain, we think about this kind of solid pink mass. But in fact, if we could look under the microscope, we could appreciate that, especially in the developing brain, there is constant amoeboid-like movement of cells reaching out and making contact or separating from each other in response to cues from the environment. Now, plasticity itself is actually a double-edged sword for humans, this prolonged plasticity. On the one hand, being able to be changed by the environment for such a long period of time may increase our risk for a number of mental illnesses associated with early life stress, for example. So I'm plotting here age on the x-axis and a number of mental illnesses associated with emotion regulation difficulties. And in red is the peak age of clinical onset. And what you can see is that these are all developmental conditions. They all seem to have their peak age of emergence around this transition between childhood and adolescence. On the other hand, this incredible plasticity that humans enjoy is the basis. It affords us this incredible cognitive growth that humans experience. We can learn from our unique environments. And to do that takes a good bit of risk taking. And the developmental research has pointed out that there's this very interesting tension during development between our incredible drive for cognitive growth and the associated risk taking, and our incredible need for security early in life. So these two things keep each other in check. So we can appreciate, for example, that this young child is able to explore his environment and take risks for learning precisely because of the presence of the species expected stimulus, his caregiver. So in other words, one way to think about parents is this strong attachment relationship affords us the luxury of risk-taking and exploration and the associated cognitive growth that comes along with that. Now, in maturity, our central nervous system plays a large role in ensuring our safe, independent exploration of the environment. And today I'm going to be focusing on the amygdala and its connections with the medial prefrontal cortex in their central role in mature emotion regulation, with the premise that the caregiving relationship plays a central role in establishing the neurobiology of emotion regulation. Now, I'd like to share with you one of my favorite quotes by Bowlby, who first wrote about attachment theory. And he points out that in the countryside in springtime, there's no more familiar sight than mother animals with their young. So familiar are these sights, and so much do we take it for granted that lamb and ewe will remain together, that we rarely ask, first, what causes these animals to remain in each other's company? Or in other words, why do we so strongly affiliate with or attach to our caregivers? And the second question is, what function is fulfilled by their doing so? Now, obviously, caregivers provide safety and food and shelter, but at a psychological level, we're going to talk about the fear and safety learning that they support as well. So let's appreciate for a moment young in altricial species. Now, altricial species refer to species that, when born, require somebody else to take care of them. These species, shown here on this slide, have one survival strategy when they're young. Their only survival strategy is to attach to a caregiver. They cannot fight and they cannot flee, so they attach. And you can see, no matter what species we're talking about, the young will follow their caregiver, which I think is very, very interesting. We didn't have to be that way. We could have been like a different type of species, like sea turtles that never meet their parents, but we stick to our parents. So early work that investigated this type of behavior established that there's a very important sequence of events for the development of affiliation or attachment-related learning versus fear learning. Namely, for a young animal that has to learn an attachment to survive, affiliation learning must precede fear learning. So, for example, what you can see here with this puppy, when he is first exposed to a human handler early in life, he will automatically exhibit an affiliation. However, if the first exposure occurs later on in this puppy's life, he will then exhibit fear. And this ordering makes sense because fear learning is going to interfere with affiliation learning. And we don't want that to happen in an animal that must affiliate with a primary caregiver. Moreover, this affiliation is seemingly obligatory because it will occur even if the animal is experiencing shocks during that affiliation learning. So in other words, altricial young are going to affiliate with a caregiver regardless of reward versus punishment. Now, more recently, there's been work replicating this effect in rat pups, and this work uses a fear conditioning paradigm. So you may remember Pavlov's dogs, where he described what conditioning was. So we're talking about the same type of process here. So in rats, animals can learn that a peppermint odor, which means nothing to them, is paired with a reward. Now, if this peppermint odor is then placed on one arm of a Y maze, young animals, whether they're 8 days old or 12 to 15 days old, will show a preference for that peppermint odor. And that makes a good bit of sense because it had been rewarded. Where things start to get a little more counterintuitive, perhaps, is when we look at fear conditioning. So if instead that peppermint odor had been paired with a foot shock, the older animal will do what you and I might do, which is we would avoid that peppermint odor. But at younger ages, this rat pup will actually approach that peppermint odor. And that may seem counterintuitive, but this same behavior has been shown to underlie the animal's ability to form an attachment to its primary caregiver, regardless of reward or punishment. Now, it turns out that if we now look at these older animals who have learned who their caregiver is, the presence of the caregiver does something fascinating to these animals. So I'm going to focus on these older animals. Oops, sorry about that. So these are the data I just showed you where they're avoiding that peppermint odor, which had been paired with a foot shock, just as you and I would. However, if they learned this association with their mother present, these older animals now revert back to that infantile behavior. They will now show a preference for this peppermint odor, which is counterintuitive. And the reason has to do with amygdala development. So here's rat age on the x-axis, and there's this period during the rat's development when the presence or absence of the mother, and I'm saying mother here because we're talking about rats and mothers are the only ones who raise the young. The presence or absence of the mother can control whether the amygdala is active or whether it's shut off. So when the mother is in the nest with the animal, when she's close by, she keeps stress hormones in the young low, and that keeps the amygdala quiet. And when the amygdala is quiet, you get this preference learning. However, when the mother is out of the nest, those stress hormones are free to circulate and the amygdala is free to mediate the learning. And so therefore you get this avoidance learning, just like you and I would show. So in other words, the mother is essentially turning on or turning off or buffering amygdala activity in the young. And this is a good reminder that the young brain is designed to coordinate with parental input. So we went on to ask whether humans are doing something similar. We would assume that they are because humans also have to attach and learn cues associated with their parents. So we brought in preschool-age children and we presented them with another fear conditioning paradigm. They would see a blue square and it would be paired with not a shock, but a terrible loud noise like nails scraping down a chalkboard. And that was called the CS plus, the conditioned stimulus. And then the triangle was never paired with anything. That's the CS minus. So children learn this association either alone or with their parent present. The parent's not doing anything, they're filling out paperwork, they're not talking with the child, but they're physically present. So after children learn these associations, we take parents away, and then we put children into a human Y maze, and we put the nasty blue square on one side and the triangle on the other, and we ask children to enter the maze and then pick a door to retrieve a prize. We show them it's the same bucket of prizes behind both doors, but we just want to know if they are showing a bias towards one door versus the other. And what we observe is that when children learn this association, this fear association alone, they do what you and I would do. They avoid that nasty blue square. However, if their parent was present during the time of learning, parents seem to be buffering the fear learning in their children, just as has been shown in the rodents. Now, to ask if parents are doing this by way of amygdala buffering as well, we brought children into the MRI scanner, and here we showed them about 30 seconds of pictures of their own parent. And then we would swap that out and show them 30 seconds of somebody else's parent, so an unfamiliar adult, and we would repeat that. And when we look at amygdala activity, during childhood, we see that amygdala activity is going down when children look at pictures of their parents. So this, again, looks like in humans, parents are buffering the amygdala activity of their children. Now, note, we lose that effect by the time we get to adolescence. And that's not to say that parents are unimportant during adolescence. They certainly are. But it may mean that at the level of the amygdala, parents have started to lose their efficacy as children start to move towards independence. And presentation of parental cues is momentarily strengthening those connections between amygdala and medial prefrontal cortex. So the buffering data I've shown you so far, whether rats or humans, requires that the parents themselves are regulated. So the parents are calm and regulated in this study. The rat mother was calm and regulated. She was, in fact, anesthetized. But we know from a large clinical literature that as effective as a calm parent is in buffering their children's stress and related neurobiology, a dysregulated or anxious parent can be just as good at amplifying or magnifying children's distress. And so to study this in the laboratory, we performed another fear conditioning study, but here we didn't fear condition the children, we fear conditioned the parents. So here, for example, I'm getting fear conditioned to this pink circle. Every time I saw it, I would hear that horrible noise and I would flinch. And we would videotape me learning this association. And then we would show that videotape, for example, to my daughter. And she would see a video of somebody else's parent getting fear conditioned to a different stimulus. And then later, we would ask her and her amygdala what she thought about these two stimuli. And we found that children's amygdalas were more reactive to the stimulus that their parent had learned to fear. And children learned to dislike both stimuli, but they learned better to dislike or fear the stimulus that their parent had been fear conditioned to. So taking this all together, what we think is going on is here's development on the x-axis. There's this period early in life where parents have a lot of ability to modulate activity of the amygdala and associated fear behaviors, both potentiating or magnifying children's fear, but also regulating or buffering it. And in some ways, we can think about the parent as having some privileged access to these deep emotional learning systems in the brain. And the idea is that what's happening during this time is important because it gives rise in part to the various individual differences that we see emerging during adolescence and adulthood. Now, with this framework, we can better start to understand the development of individuals who may have experienced caregiving-related early adversities. So I'll be referring to this term CREES for the remainder of my talk, for maybe 5 minutes or so. And when I talk about CREES, I'm referring to extreme adverse experiences that children can have involving their caregiving relationship. And CREES can include abandonment or permanent separations, abuse or neglect, multiple caregiving switches, and or institutional care. Now, if we look at the development of emotional behaviors following CREES, we and many other groups have found that there is a significant increased risk for behaviors like anxiety and depression relative to individuals who've experienced typical caregiving, CG is caregiving here, as well as challenges to conduct problems and attention. So these are large effect sizes. In fact, early caregiving adversities is the #1 environmental risk factor for these behavioral problems. However, at the same time, as big as that risk is, there are a lot of individual differences. So each dot I show you here is a child, and you can see there's a large spread So a lot of our work is focused on trying to better understand what differentiates a child who really struggles, for example, in conduct problems versus a child who is not. And some of the neuroscience can give us insights into these types of questions. So if we look at amygdala and prefrontal development following CREES, for example, in response to fear faces in an MRI scanner, it's common to see amygdala hyperactivity following CREES. And that's very consistent with the hypervigilance and tendency towards anxiety and depression that can follow early life caregiving adversities. And we see atypical development of connections between the amygdala and that prefrontal cortex, which is going to play a large role in independent emotion regulation at older ages. And we hypothesized that altered amygdala development following CREES might place limits on parental buffering of the amygdala. And so we repeated that paradigm, that experiment I described earlier, where children saw pictures of their parent and also pictures of a stranger. And these are the data I showed you earlier. We're following a typical caregiving history, which just means that there is an absence of CREES. We see that buffering of the amygdala in childhood. However, following CREES, we don't see that same buffering. At the group level, we see evidence consistent with the idea that there are limits on parental buffering of the amygdala following CREES. However, these bars here are error bars, which means there's a lot of individual differences. So some children following CREES are not showing buffering of the amygdala by their parent, but some are. And so we interrogated those individual differences longitudinally over a number of years to see if that made a difference in terms of mental health outcomes. So if I can draw your attention to the right side of the screen here, I'm plotting anxiety symptoms following CREES. So at time 1, we can see that following CREES, there's a significant increase in anxiety relative to a comparison group of children who did not experience CREES. But we split these participants into those who showed amygdala buffering by their parent at time 1 and those who did not. They didn't differ at time 1. But when we brought them back in 2 or 3 years later, we see that there is predictive value in what amygdala was doing to the parent at time 1, such that in the solid line, children who exhibited amygdala buffering at time 1 had significant decreases in anxiety symptoms over a 2 or 3 year period, so much so that they are no longer statistically different from children who did not experience CREES. In contrast, those children who did not show parental buffering of the amygdala at time 1 maintained those higher levels of anxiety as they transitioned into adolescence. And you may be wondering, as we did, what's the difference between these two groups of children? And what we observed was the attachment relationship between parent and child seemed to really matter. I should mention in this particular study, the parents we're looking at the time of the study were all adoptive families. And children who expressed having a stronger attachment relationship with their parents were those children who showed amygdala buffering at time 1 and those who showed significant decreases in anxiety at time 2. And this finding is very consistent with many in our lab and others, suggesting that stronger family relationships, so here on the x-axis is the child's feeling of security in the family, is associated with lower levels of anxiety and depression. Now, these are really important data, but this is a correlation, which means we can't really know from these data what caused what. So maybe it's the possibility that children who are lower in anxiety and depression are more easily able to form these strong relationships with their parents. And that's where we need to use experimental designs. And so I've been fortunate enough to collaborate with Mary Dozier, who's at the University of Delaware, and she developed an intervention called the ABC intervention. It's the Attachment and Biobehavioral Catch-up intervention. And she administers this intervention through random assignment to families who are at risk for maltreatment. And the intervention is quite brief, but its goal is to strengthen the attachment relationship between parent and child. And she's followed these families for over a decade now, and she's found some, I think, incredible results from this intervention, such that children who were randomized to the ABC intervention were more likely than those in a control condition where families received psychoeducation about the importance of families. Children in the attachment condition were found to be more securely attached to their caregivers, have healthier cortisol production, the stress hormone cortisol, develop better impulse control, control their anger better during a challenging task, and have improved executive function. So I approached Mary Dozier's group and I said, it would be really amazing if we could start to collect MRI images to know if this early intervention actually had an impact on prefrontal and amygdala development. And so what we observed is, in fact, there was an effect of this intervention. As you can see here in orange, I'm showing you areas of the brain that were more engaged in the individuals with the ABC intervention. And I'm pulling out those data and plotting them here, showing you that the prefrontal cortex was more active in the ABC condition than those individuals in the DEF condition. So these data have been very important for us because they show a causal link between early caregiving experiences and these later brain development in these later brain regions that we know are important for emotion regulation. So to conclude, we know that human emotional brain development is very slow, but that there's value to this slow pace. It gives us a very long time for learning about emotions and how to regulate them at the behavioral and brain level. Systems involved in emotional learning and reactivity, like the amygdala, seem to develop early, but those that are involved in regulation, like the medial prefrontal cortex, seem to develop later, well into adolescence. So we can think about caregivers, the primary caregivers or parents, as being this important external regulator of amygdala medial prefrontal cortex development, having some privileged access to those deep brain regions. And caregiving related early adversity does significantly increase the risk for altered amygdala cortical development, but there are large individual differences that are important to study, and we're only at the beginning of being able to answer some of those questions. And finally, strong parent-child relationships always have very powerful effects on brain development, but perhaps especially so in the context of children who have special needs. So with that, I would just like to acknowledge those that have contributed to this science and thank you for your attention. And I'm happy to take any questions.
Wow, Professor Tottenham, thank you so much for that. That was a lot of information. I felt my emotions being unregulated because I was thinking, how did I raise my own kids? So there are a lot of interesting questions in the chat, and we'll try to get to as many as we can. Again, if you have a question, please put it in the Q&A section. Let's see. Catherine Sheehy from class of '03 and law school '06 asks, can you talk more about caregiver switching qualifying as creeds?
Yes. So the switching, as we define it, is the situation that's commonly observed, for example, in foster care arrangements. If a child is unable to stay in a home and has to change attachment figures completely, we get very concerned about that because that's a major disruption in the integrity of that caregiver child relationship. So I'm not referring to things like joint custody that families may share, or um, daycare situations, but really thinking about those permanent changes in household that children experience that come along with a whole host of changes in the child's life.
So other questions do deal with whether separated parents have an impact on early brain development, as well as the type of care, daycare, having a nanny, or having the parent there. Is there I don't know did you want to take that one after another, or
Yeah,
is there any
they're a little
separate
bit
research that
different.
deals with that? Yeah.
Yeah. I mean, in the case of daycare, we know that there are a lot of benefits from daycare, and teachers are not the same thing as parents, but they're really good adults for children to get to know. So typically, what happens is in a child's life is they form these attachments to their primary caregivers, and then they learn how to generalize that relationship to other adults. And children will certainly learn a lot of important skills being separated from their parents, and then reuniting. So I remember when my daughter was in preschool, there was a mantra that mommy always comes back. And that's a really important lesson because we won't always be around our parents. And we have to learn that separation doesn't mean permanent separation, or hopefully it doesn't mean permanent separation, but it means this is your time to go off and explore the world. And at the end of the day, you're going to come back home. So that is generally what the science supports, and is consistent with the neuroscience. In terms of families that have separated, it's a little bit hard to answer because everyone's family's situation is a little bit different in terms of how custody works. But what we know is children can form multiple attachments to, one parent, they have one type of attachment relationship, and the other parent, they have another type of attachment relationship. And that's true whether the two parents live in the same household or not. And in fact, what we're learning is the model of thinking about a single attachment figure is probably not universal. And depending on family structure and culture, the attachment figure may be comprised of multiple people. And really, what we're learning through that attachment relationship is, does the world have my back or not? And that could be taught from one person, or that could be taught from more than one person. So if we think about extended families, for example, if you live with your grandma and your auntie in the same house, I mean, this idea of how we live, I'll just say, in our weird societies is kind of new for human society. And so we really have to be flexible as infants. We have to be flexible in learning who our caregivers are. And in fact, it's one of the things that I didn't talk about today, but is really fascinating about humans. We're a species that alloparents, meaning we take care of others young. And that's been true throughout human history. So I think we need to appreciate that the infant's brain is wired, is expecting to be able to have that type of flexibility with, learning who's important in its life.
So it sounds like the old adage of it takes a village. That actually gives the kids more chances, right, to form these attachments and have very positive parental figures, if not their actual parent.
Exactly. And give not only flexibility in their understanding, but also literally provide more people who can provide care.
Yes. I wanted to ask also about screens. Uh, parents worry about them a lot, whether they have their kids looking at screens. But there's also, parenting now is hard, and parents are also always on their screens. So how does that affect the research when parents are present, but they're not always there? It's kind of just part of modern life now.
Yeah, it's a really good question. And I have not personally studied screens, but you raise a good point about, I think the term is presenteeism. It
Hmm,
means when you're physically present, but your mind is not there.
interesting.
And so, you know, I think that the parent's physical presence matters a lot. If you think about taking your child to the pediatricians, for example, you never just send them in on their own. They probably wouldn't go, but you go in with them just because your physical presence is doing so much to them physiologically. But if all you're doing is being present, and you're on your screen the whole time, then you're not also teaching your child that I'm here for you. I'm actually an agent that can help scaffold you. And children who follow their attachment figures are going to follow everything their attachment figures do. So
Hmm.
it's also presumably influencing the future behaviors of that child. But the other thing when it comes to screens, I don't know, what the most cutting edge research is on screen time. But I can say, based on this research, that the one thing I would be mindful of is there something that screen time is taking the place of that could be a good learning opportunity? That being said, we also have to appreciate that an exhausted parent is going to have a very difficult time always being the parent that they want to be. And so we don't want to put so much pressure on parents to be the perfect parents all the time, because no one can handle that. So really finding a balance between what makes you feel fulfilled in this life and, sort of in your own skin is probably the best thing to do for our children.
Thank you for that. I'm feeling better. Okay. Another question, can Cree's damage be reversed or mitigated in adolescent years? How?
Yeah, so that's a great question. And if I had more time, you can invite me back one day. I'll share more data. We and many other labs have data now showing that there does seem to be some really ameliorative influences of intervention during adolescence. And this can take various forms. My read of the literature is the most impactful interventions are going to be around intimate social relationships. So whether that's with peers and romantic partners or with the parent. And that's consistent with the data that I ended on, showing that these strong parent-child relationships are seeming to have a really big impact on turning the dial for children that experienced really difficult things early in life. And so that is actually much of the work going on in our laboratory right now.
Oh, interesting. So sometimes when they say, like kids who had a hard time or something traumatic happened, but there was a coach or a teacher or an uncle who, really took an interest in them and took them under their wing as adolescents. So that could mitigate the effects of Crease.
That's right. And the formal term for what you're describing is resilience factors. And so, for all of us, no matter what our early life experiences are, finding sources of resilience in the environment are what kind of buffer. I used, the description of parents as buffers, but buffer some of the impact of what could turn into toxic stress situations and make them more manageable and allow for better coping.
Another interesting question. I wonder if kids who have immigrant parents and are sent back to their parents' home country while their parents work in America would exhibit any differences in amygdala reactivity or connection to the frontal cortex.
Mm-hmm. It's a great question. This is a common practice for many families. It just makes sense for a lot of families. Um, Surely there will be a lot of differences in what families are doing, but often children are sent to live with grandparents, for example. And so this is another example, a long distance example, I guess, of the extended family that I described earlier. And so a lot of what happens is going to depend on the age at which the child moved, as well as the quality of the relationship with the, let's just say, grandparents that the child lived with.
Um, Sean Lander says, thank you. This is a great compliment to your presentation at Reunion in 2024.
Thank
I'm
you.
curious to know whether there is parallel research in older adults, particularly those with relevant dementias, to understand whether there are reversions to childhood amygdala patterns in adults who didn't have CREs, but perhaps not in adults who did, such as migrants, refugees, war and genocide survivors.
Yeah, it's a wonderful question. I don't know the answer to it, but I do feel confident saying that as humans, what are we? We are social creatures, and our brains are wired with the expectation of collaborating with other brains. So I often like to think about our eye contact as being a big synapse between two people, or holding hands as a big synapse between two brains. And so, Sean, I think your prediction is right, that we would expect really important buffering, stress reduction for individuals that are struggling, regardless of where they are in life.
It's good to know for many of us who are also doing elder care
Mm-hmm.
too. Um, Nicole from the college class of 19, how are parents better prepared today than in previous generations?
That's a good question and a hard one to answer because there's so much variety in parenting behaviors, no matter what era we're talking about. But in broad brushstrokes, I already talked about one difference, which is the way that we structure our homes and our families. We can now leave very often the town that we grew up in. We don't live very often with kin. We can, but it's becoming increasingly popular for people to live far away from kin. And so you change the family structure, which changes the way that people parent, sometimes for the better, sometimes for the worse. It can add more stress, it can decrease stress depending on your family's structures. But I also think that parents just have a lot more information today. And the mere recognition, actually, that children require special or different behavior than adults is a very modern concept. It really was, following the Industrial Revolution that people started to recognize that children aren't miniature adults, and they may maybe we should treat them differently. And it wasn't really until Freud that this idea that early experiences matter for later outcomes even got onto the radar. So all of these things we're talking about today are really the products of recent history. So we have so much more information today, which I think is a double-edged sword. I think it can, we want to be careful that it's not making us anxious as parents. Sometimes I'm asked as a parent myself, am I more anxious because I know about these types of effects? And actually, I would say I try to make me less anxious, because I recognize that really what we're talking about is patterns of behavior over time. That's how we learn in any relationship, right? If someone does if as a parent you make one mistake, probably it's not going to have major impact. It's really kind of the routines. It's, what we do day in and day out. So anyway, I would say I would just be mindful of parents becoming too anxious. And then the last thing I'll say is, again, I'm speaking in really broad brushstrokes, so I feel a little uncomfortable making generalizations, but arguably parents also have added tasks that maybe previous generations were less likely to have. So for one, we may have different aspirations for our children. So anyone who's tried to get their kid into college, right? Thinking about what that process is like, that's a lot of pressure. But more what I mean is our period of being juveniles as a species continues to get stretched out. So adolescence is getting stretched out further and further into the early 20s. It wasn't so long ago, and maybe and there surely are families where this is still true. Once you hit 18, you were independent, you had to move out, or in another culture it might be even younger ages. So we're supporting children for a much longer period of time too. So not to mention everything else we have going on. So I think that parents are a lot smarter and there's a lot more to do, but I would just caution to say we should also kind of not be too hard on ourselves as parents.
Um, yes, I'm trying to internalize that now. All of this. We have one more question in the chat. You mentioned that the age of when a child moves to a different set of caregivers, like grandparents, uh, matters. What impact does age have, and are there recommendations for when such events are better handled by children? I think for your research, like 10 is a really important age, right? Are there kind of any other ages that are these like watershed moments?
Yeah, I don't know the answer to that exact question, because I don't know about the research of uh, the example you gave of children who might move countries but stay within the family. I'm going to look it up after this. Thank you. Um, but the ages that developmental psychologists pay a lot of attention to is, for example, that first year or two of life is the time when children are learning who their primary attachment figures are. Actually, there's, I think, a very interesting set of findings if you look across species. That period when we learn who our attachment figures corresponds roughly to the age at which children start walking. And if you look across species, that tends to be true. So in animals like ungulates, like sheep, they start walking in about an hour. So their period of learning and attachment is that first hour. So, in other words, the take home is that attachment learning seems to have evolved for the young altricial animal to learn to always stick to the parent. Even once independent locomotion comes in, they will continue to come back to the parent. So, it's important to think about those kinds of milestones when making decisions about children's lives in terms of what they're learning and who they're learning to stick to. And once they've stuck, how then do you introduce new important people into their lives in a way that makes them feel safe and secure?
Professor Tottenham, this has been such an interesting conversation. We will absolutely have you back because we have more questions. Thank you for sharing your scholarship and research with the community, your community, college community, and a big thank you to all of our alumni, students, and friends who joined us. We have more alumni talks and big topic short takes. Please join us. There's one tomorrow at 11 a.m. Eastern Time. It's Stories of Race and American Sculpture with Smithsonian curator Karen Lemmy, class of 95. And then on November 19th, with Professor Rishi Goyal, Director of Medical Humanities, we have Medical Humanities, where two cultures meet and flourish. If you have ideas for other topics related to wellness, or any other topics, we'd be very happy to hear them. And we'd also love for you to join any upcoming programs. Bring along other classmates and for you to volunteer your time and expertise like Professor Tottenham has. If you'd like to get more involved, please reach out to us at ccalumni@columbia.edu. And again, we'd love to hear all of your ideas for topics, future topics. So thank you, Professor Tottenham,
Thank you.
for spending your time with us. And have a great afternoon, everybody.
Bye.
Bye.